And welcome everyone to NSTA Engage Spring 21. My name is Heather Ryan and I'm this session's moderator. Chris Herman and Rachel Mosey are supporting us behind the scenes today. And thank you for joining us for CRISPR as an adaptive immune system in bacteria. Tim Herman will be our presenter for the next 45 minutes. Please use the chat to communicate during the session, and the chat will become part of the recording that will be posted on the Spring Engage site tomorrow. Rachel will post links to the resources, registration for your free student modeling packs, and session evaluation in the chat throughout the session. I will also monitor the chat for your questions. And we will let you know when you can unmute and interact. It is now my privilege to introduce my father, Dr. Tim Herman. He earned his Bachelor's of Science in Chemistry from the University of Nebraska, his PhD in Biochemistry from Oregon State University, and then pursued postdoctoral studies in molecular biology at Harvard Medical School. Tim then joined the biochemistry faculty at the Medical College of Wisconsin, where his research areas range from the synthesis of chemically cleavable biotin-labeled nucleotide analogs to the development of novel approaches to structure-based drug design. After almost 20 years of research, teaching graduate students and medical students at the Medical College of Wisconsin, Tim moved to the Milwaukee School of Engineering and founded the Center for Biomolecular Modeling to develop instructional materials, provide professional development, and deliver outreach programs. Shortly thereafter, he founded 3D Molecular Designs to make these models, kits, and materials available to teachers and students. He has spent the last 20 years developing molecular models, kits, and communicating fascinating molecular stories to educators, students, and the public. His efforts now reach more than 1 million students a year around the world. Now we'll begin the program. Go ahead and take it away.
Okay. It's always a very uncomfortable beginning to these sessions, but thank you, Heather. So yes, I'm Tim Herman, the MSOE Center for Biomolecular Modeling. If you don't know about us, let me just quickly say that we're all about proteins. We use 3D printing technology to make physical models of proteins, and then we tell molecular stories. And we try to tie this back to those foundational concepts of molecular biology that all of you are teaching. So we're fortunate to have funding right now from the NIH SEPA program. That's a Science Education Partnership Award. So the current project is called Science and Ethics of Genome Editing. And we're talking about CRISPR-based genome editing. So this protein I had out here, I'll tell you what this is. This is CRISPR-Cas9. So we will get to this actually not so much in this first workshop this morning, but in the second workshop, we'll focus on how this Cas9 endonuclease actually works. What we're going to do in the first workshop here in the next 45 minutes is talk about what we refer to as CRISPR biology. And what we mean by that is how does CRISPR, this CRISPR system, function as an adaptive immunity system in bacteria. So we're only going to be talking about where this system evolved in bacteria. And then in that second session, we're going to focus on the Cas9 enzyme and how we're beginning to modify that naturally evolved protein or enzyme to be an even more effective tool to begin to edit the human genome. So that's where we're going both in this session and one that immediately follows this. All right, so if we have the slides up.
Yes.
Is it up now?
There you go.
All right, so this is one of those iconic slides. If you've looked into CRISPR, you very possibly have come across this. I don't remember exactly what paper this comes from, but you'll find lots of schematic illustrations of this system on the internet. Great time to be a teacher. You've got all those free resources out there as close as a Google click. So let's just go through this quickly. This is a bacteria or a prokaryotic cell. Let me find my mouse here. And this gives a nice overview. When you understand CRISPR biology, then this makes a whole lot of sense. Don't allow yourself to think that all you have to do to teach CRISPR to your kids is to show them this and to spend 5 or 10 minutes walking through and explaining what's happening here. That's an easy way to teach. It's a very efficient way to teach. You could teach CRISPR in about 10 or 15 minutes. Unfortunately, I think you'd be fooling yourself if you thought that that really made a great impact on kids and they're going to remember this for very long. So I'm going to use this just to sort of set the stage here. As teachers, you probably have an idea. Maybe you know a lot about this CRISPR system, in which case this makes sense to you. But let me just hit the high points here in about 2 minutes. So here's a bacteriophage that is infecting this bacteria, and it's inserting its red genome into the bacteria. So this bacteria has a CRISPR system. So it has evolved a complex of enzymes that can go out and detect this foreign incoming double-stranded viral DNA. And it can cut out a small section of that is a very specific length. And then it can bring that small section of double-stranded viral DNA back, and it can insert it into a region of its genome, which we refer to as the CRISPR array. And it's just an array of viral sequences, the colored sequences here in squares. It's an array of them separated by these black diamonds, which is a repeat structure, not a repeat spacer. We have repeats and we have spacers. I should have separated these two words a little bit here in this graphic. But anyway, this is what happens. So you get a little piece of the red DNA, you put it here, and then that enables you from that moment on to generate this guy here, which is a CRISPR-Cas9 enzyme, an endonuclease, which is now programmed with a piece of guide RNA that includes this red insert, this red spacer. And this programmed Cas9 then can float around in this bacterial cell, constantly on guard for another red bacteriophage virus that is trying to infect it. And if another red virus comes along, then this surveillance complex can bind the double-stranded viral DNA and it can cut it. All right, and in that way, it prevents subsequent infections by any bacteriophage that it has encountered in the past and successfully integrated a piece of that viral genome into its CRISPR array. So CRISPR is this wonderful acronym. And I hope none of you have seen John Oliver's treatment of genome editing recently, because John Oliver has his own interpretation of what that acronym stands for. And I'm not going to even repeat it here, because then you'll, and I shouldn't have even said this, because now you're probably all Googling on your phone, John Oliver CRISPR. But what CRISPR really stands for is clustered, regularly interspersed or interspaced, short palindromic repeats. And on one hand, it's a horrible acronym that kids in the beginning are going to find really, really confusing. But I actually love this acronym. Once you understand this story, this makes all sorts of sense. You see these black diamonds here? Those are short palindromic repeats. Okay, and notice how these short palindromic repeats are all clustered together, and they're interspersed by these spacer sequences, all of which have the same length. So they're regularly interspaced short palindromic repeats.
So we're starting to get some questions in here. So
Okay.
Let's move on up to how CRISPR was discovered, and then it's going to make sense about how we can talk about acquiring and all of those next items.
All right, what we really want to do today is get out this, what we call the adaptive immunity kit, and we want to go through that kit with you. But I don't think you should just get that kid out and say, all right, students, today we're going to talk about CRISPR and you're going to use this model to understand it. I would suggest that you start with an activity and you ask the question, who discovered CRISPR? And then you say, wait a minute, that's not even a good question. A better question is, how was CRISPR discovered? Okay, and here's the beginning of that answer. How was it discovered? It was the first report of a CRISPR system occurred back in 1987, right up here, by a group of Japanese researchers. Ishino was the lead author here at Osaka University. And these people were studying a gene in E. coli, a gene that encoded alkaline phosphatase. And they wanted to, they cloned the gene and they sequenced it. And they were trying to understand how the gene was regulated and so forth. Back in 1987, you could publish a credible paper in which you simply reported the nucleotide sequence of a gene. Things have changed immensely since that time, but that was this paper. You won't read anything about CRISPR or repeated sequences in the title or even in the abstract of this paper, because that really wasn't their focus. But in the very last paragraph of the discussion of this paper in 1987, this is what they said. They said an unusual structure was found in the 3' end of the IAP gene. 5 highly homologous sequences of 29 nucleotides were arranged as direct repeats with 32 nucleotides as spacing. And then they go on to say down here, so far, so that was what they noticed. They noticed this unusual pattern of sequences. And so far, no sequence homologous to these has been found elsewhere in prokaryotes, and the biological significance of these sequences is not known. So this was what happened in 1987 that began a almost 30-year-long effort by people all around the world to understand the significance of these CRISPR sequences, these CRISPR arrays. So the question, this is often how science happens. Yeah, the question you pose to your students is, could you have discovered CRISPR? So you want to get your kids to imagine if they had been a junior member of that Japanese research group in 1987, and your group successfully cloned the gene, they sequenced it, they went a little bit beyond the end of the gene, and then they were writing this paper about how the gene was structured and regulated and so forth. And the question is, could your students have recognized the pattern that they reported at the very, very end in just kind of a throwaway paragraph of this paper? And you've all received a student modeling pack, I believe, with these colored nucleotides. And I think,
or at least some of them, we hope that some of you
Okay.
have, and the rest of you, we're happy to send you a pack. Make sure you register the link, Rachel.
okay, so if you have, or if you can get a hold of these foam nucleotides, color-coded, and if you join them together, 5' to 3' in this particular sequence, this is the sequence that the Japanese group noticed and reported. So this is one of those direct repeats that make up or define a CRISPR array. So I hope you have that in front of you. And if you do, then if you look at it, can you see the pattern? And what I suggest you have your kids do is you ask them, you give them this sequence in one of several forms and ask them to look at it, to think about it. So here's another form that you could give it to them in. This is just a
Hold it so
long-
it faces you.
Hold it so it faces me over here. All right. Here's the 5' end, G, G, A, G, T, T. And it goes on and on and on. I forget exactly how long this is, maybe a couple hundred nucleotides. And you should give this, you could give this to your kids and you could say, could you have detected the pattern here that the Japanese group detected? And I would bet you money that the answer would be no, because it is virtually impossible, I believe. unless you have a really, really interesting mind, to actually see the pattern here. So that wouldn't be very effective. So let me show you something else you could give them. You could give them this, which I really like because this is a DNA trace. This is a sequencing trace. So it begins to suggest to kids how this sequence was determined. Yes, it's a sequence of nucleotides, but here's the actual data that determine that sequence, which is printed across the top here. I'm sure you can't see this. But what we've done here, we've actually made it too easy for students because we have indicated in these pink blocks the repeated sequence. So there's the pattern. And even without these pink blocks, with this visual representation of the sequence, I think your students would have a chance then of being able to see how this pattern of black and blue and green and red spikes, peaks, is repeated here and here and here and here. So these are the repeats that were noticed and first reported in this paper. So the easier way, a better way, I think, that you might want to expose this to your kids is what Heather is going to show you right now. So on the screen, I think this is a word-wrapped version of that sequence that was determined. And if you were to just look at that right now, I think you would still be hard-pressed to say, aha, I see some regularly interspaced short palindromic repeats. But now Heather is going to take her mouse and she's going to begin to change the margin over there on the right. And I want you to watch, I'm going to give you a clue here, watch the left-hand end of that sequence, those aligned sequences. And as Heather shortens the length of the lines, see how those green sequences and now the red A's at the very, very left-hand part of the page. So when you get right there, when you word wrap this long continuous sequence, at this particular point, if you have color-coded the nucleotides, then these repeated sequences pop right out. You see the A's, you see the C's, you see the G's. Now I will make one point here, and that is they're not perfect repeats. You can see that there are some nucleotides in the middle there that don't match entirely, and that's because this is biology. And nothing in biology is, I shouldn't say nothing or always, but there's always some sort of ambiguity in biology. And you have to be careful with that because sometimes kids react very negatively to that. But the thing I want you to think about here is the only reason this text wrapping trick works is because the length of the repeats on the left and the length of the sequences that provide the spacer between the repeats, both of those elements are of a specific length. So in this particular CRISPR system, the short palindromic repeat is 29 nucleotides, and the spacer sequence is 32 nucleotides. So yeah, in the next slide, then we've just changed the color coding now so that we're not focusing so much on the repeat. All of this is in black. But now we're focused on these spacer sequences. And as it turned out, this is what the first big step in recognizing or understanding what the system was doing was the recognition that these sequences, these spacers, are homologous to different viruses. And in particular, they represent sequences from a number of different bacteriophages that had tried to infect this bacteria in the past. So again, and they're in chronological order. You know, you look at the first one in a CRISPR array, this is going to represent a sequence homologous to the virus that most recently had an encounter with this bacteria. All right, and yeah, there's the 32 nucleotide. All right, and then we'll just go one step further here and show you if you now stretch all of this out in one continuous sequence from 5' to 3'. You have these black repeat sequences of 29 nucleotides separated by these colored bacteriophage spacers, okay? So those are clustered, regularly interspaced short palindromic repeats. So I would recommend that you engage your kids in some sort of a challenge, looking at those sequences in one form or another, and ask them if they can notice, if they can detect this pattern. And if so, they can be reassured that had they been in that Japanese research group in 1987, they could have discovered CRISPR. All right. And
Okay, you have 2 minutes if you wanted to do any of the hairpin stuff
all right.
there.
All right. So I have a little bit of time here.
Just 2 minutes though.
So, we always tell teachers, don't teach the what of science, teach the why of science. So the what is that there are these short palindromic repeats in a CRISPR array. So the question you would hope your students would ask is, why? So what's the significance of this repeated sequence being a palindrome? And I can tell you right now, it doesn't have anything to do with restriction enzymes that recognize palindromes and cut. That might be their guess, it'd be a good guess, but they would be wrong. But anytime you have a sequence which has some palindromic nature, and these are also referred to as dyad symmetry or twofold rotational symmetry. Anytime that happens, then it gives you the possibility of doing this. You see these C's and you see these G's. You can form a stem loop structure or a hairpin in which all of these G's base pair with all those C's. And then here we have an AT base pair. And
Slide it up or turn it sideways a little bit.
okay, yeah. So here's a hairpin, here's a loop down here. And of course, this happens on one strand of double-stranded DNA. The symmetrical thing is happening or can happen on the other side. So what am I suggesting to you here? I'm suggesting a possible answer to the why. And I don't know for certainty that this happens, but I think it's a question you should explore with your students. I think you should certainly congratulate them if you can get them to ask the question, why? And remember, we'll come back to this a little later once we've gone through the kit, and I can hopefully connect this hairpin structure to what's going on in that CRISPR system. So just hold that idea in the back of your mind for a minute. All right, we're now going to turn to the kit, and let me pull these things then onto the table so you can see this. All right, so here are the basic elements of the adaptive immunity kit. And we've stamped out of foam several things. We have 3 bacteriophage up here, purple, blue, and tan in color. So I should have them in that order. Okay.
Do me a favor and pull off that tape. It's bothering me.
This tape right here?
Yes. Thank you.
Heather's very persnickety about her models. All right, so you've got, there are all kinds of viruses floating around out there just waiting to infect a bacterial cell. This is the cell wall, the membrane of a bacteria. And inside, there is, of course, the bacterial genome. And this represents just a small part of that bacterial genome. And if you look right here, we have a very short, what we're going to refer to as a CRISPR array. Okay, you already know what that means. Here are those short palindromic repeats. Okay, here are the short palindromic repeats that the kids have just explored. Here are the spacers in between. This represents a piece of DNA from this tan colored virus that tried to infect this bacteria. a couple of months ago. And then here is a bacteriophage sequence from this blue virus that had a run-in with this bacteria maybe a year ago, all right? And then what happens is this purple virus settles down and begins inserting its purple double-stranded DNA genome into this bacteria. And what's going to happen then is something that we don't really model in this kit, but there are some other genes here as part of this bigger CRISPR system that they encode proteins that can go out and they can recognize this as a foreign double-stranded DNA from a bacteriophage. And then they've evolved a system, a CRISPR system, to protect themselves. What this acquisition complex of enzymes does is that it cuts out a short piece of this purple double-stranded DNA of a very specific length. I think we said 32 nucleotides long. That's going to be the spacer that's going to get incorporated into this CRISPR array. The whole thing is called a CRISPR locus. We'll talk about what happens upstream from the CRISPR array here in just a second. But a short piece of purple double-stranded DNA is carried back to this CRISPR array. And that acquisition complex then can recognize these repeats. And this is where that hairpin structure might come into play, because if you melt out these two strands of DNA, and if each strand generates this little hairpin, that might serve as sort of a red flag to the acquisition complex that, hey, insert this purple DNA just upstream from the first repeat. It doesn't put that purple DNA just willy-nilly anywhere here. It recognizes this red leader sequence in conjunction with this repeat. And what it does, it cuts one DNA strand here, cuts the other DNA strand here. These hydrogen bonds that hold this together come apart. And then, I forgot to indicate this. This is the little short piece of purple bacteriophage DNA, 32 nucleotides in length. So it comes in, it is brought in by that acquisition complex, and it is ligated right here into the CRISPR array. And then you've got these gaps, and then an DNA polymerase comes along and extends this 3' hydroxyl group one nucleotide at a time, so it fills that in. So here our model is no model is completely accurate. We just plug these in, and your students might think, oh, you paste in some nucleotides here. And this is actually a polymerase that goes one nucleotide at a time until this is filled in. All right, that's the first step in acquiring resistance to this virus. You have successfully incorporated the purple spacer into the CRISPR array. All right, what happens next?
I'm going to ask a question, because as I'm going through this, and this is a question that we were asked before.
Okay.
And so part of the question is, I know that they're cutting the DNA, but how does this bacteria survive this infection? But I am just realizing is with cutting that small part of the DNA, that could be then disrupting the infection process of that virus. Is that correct?
That is correct.
That may be part of it?
That is correct. And these are the kinds of questions you really need to somehow teach your kids to ask. And that's not a small feat. But yeah, you want your kids not to be just writing down everything you say, but you want them to be stopping and thinking about what you're saying. So there, we could spend the next hour here just talking about those sort of deeper, sort of nuanced understanding of this. So we don't have time to do that. So I'm just going to say, it doesn't clip off the first 32 nucleotides from the end. it chops out 32 nucleotide segments from all over that bacteriophage genome. And if it happens to chop out a piece of the genome that doesn't, isn't critical to the survival of that bacteriophage, then we're never going to know that because this bacteria never survived and acquired resistance to that bacteriophage. So what we see is what works. We see in these E. coli that can live in this environment of viruses, we see those who have successfully acquired the resistance. And then your other question is, how did it survive that first encounter? Well, this isn't the only viral defense system that bacteria have. Just like we humans, we have an innate immune system that sort of has a sort of general response to foreign DNA that comes in. Bacteria do as well. One example are restriction enzymes. That's the one that your kids may be familiar with, depending upon if they've had a biotechnology course or if you've talked to them about restriction enzymes. That's a system that will recognize a particular sequence, what's the EcoR1 restriction site?
It's behind you here.
Yeah, we'll get to that. We'll get to that in the next workshop. So the point is, there are other systems that tend to suppress this initial infection, and then if the bacteria can survive that first attempt and get this incorporated, from that point on, there's very, very little chance that this purple virus will be able to infect and ultimately kill this bacterial cell. Okay, any other questions? I don't know, I can't really.
Yeah, so there's one about have phage co-evolved to circumvent CRISPR.
That's a great question. And if your students have participated in the Science Olympiad protein modeling event from several years ago, they will have constructed a physical model of a protein called anti-CRISPR, which is exactly that. Some phage can make a protein which binds to that Cas9 endonuclease that we're going to look at in a minute, and prevents it from then finding this this viral DNA that it is programmed to find. So the answer is yes. There's a constant arms race between the bacteria and the phage that are trying to kill them. So that's a very interesting part of the story. All right, let's go on and try to finish this and do the next step then. Let's look at these genes that are often upstream from there. They're always in the same vicinity. They're usually upstream from the CRISPR array. And we've only shown two genes here. There's an orange segment which encodes something called tracer RNA. So there's a tracer gene, and then there's a white gene, which in fact encodes a messenger RNA, which is translated by a ribosome into a protein. So this is a messenger RNA that gives rise to the Cas9 protein. Okay. Unlike this mRNA encoding gene, which results in a protein, when you express this tracer gene, you make a piece of RNA, which is not messenger RNA. You might make, in this case, we're going to make multiple copies of this tracer RNA, and it's a structural RNA, and we'll see what it does in just a minute here. Should also point out that you can translate this mRNA several times by a ribosome, so you can make multiple Cas9 proteins. Okay, when you express this part, like when you express the CRISPR array, and again, when I say express, your kids should realize that there's an RNA polymerase that's going to come down and it's going to express this chunk of RNA. And again, these are going to be, again, structural RNAs. These are not messenger RNAs. But what's going to happen now, here's the magic. The the 3' end of this tracer, I'm sorry, the 5' end. So you can, if you're familiar with all of our other models, whenever you see this arrow, that means this is the 3' end, so this is the 5' end. And this has evolved such that the 5' end of the tracer is homologous, and it forms Watson-Crick base pairs with the 3' end of this repeat. Now, how clever is that? And clever is the wrong word because no one designed this, but this is the way the system evolved. These two separate structural RNAs base pair like this, and then bind to this Cas9 protein, okay? And in so doing, we now have a Cas9 endonuclease programmed to search out and find a purple sequence, which is complementary to this spacer. That's the whole key right there. And at the same time, we program this Cas9 to be on the lookout for the purple virus. We can do the same thing here, and we can program this Cas9 to be on the lookout for the tan-colored virus. And finally, the blue virus here. So we express from this CRISPR locus some structural RNAs, which we're going to call then called guide RNAs. And we're going to produce then 3 different endonucleases that are programmed by these guide RNAs, sometimes referred to as dual guide RNAs. Because there's one RNA, there's a second RNA. Let me caution you about something. When you express this CRISPR array, you make a continuous piece of RNA that goes from purple to black. So that's one transcript. Don't let your kids get confused to think that you make a purple and a black and then you somehow splice those together. We only do this because we want to highlight in color the identity of these, the function of these different sequences. All right, so then that's about as far as we're going to go in this particular session. But let me just say then that all of these RNA program Cas9s then can float around in the cell watching for other bacterial, other viral genomes that get injected into the cell. And when they find it, they can then bind to that double-stranded DNA, and they can interrogate it with this spacer sequence. And if they can find some DNA that is complementary to this Cas9 endonuclease is going to make a double-stranded cut. It's going to has two separate active sites. One active site cuts the top strand, the other active site cuts the bottom strand. So it effectively makes a double-stranded cut in this invading DNA, and you have defeated the virus. Okay.
Okay,
How are we doing on time here?
we do have time for questions and to review a couple of other things. Well, one question has come up of how does the endonuclease recognize so many different spacer sequences?
Yeah.
And we're really covering that in the next session, but
Yeah.
we may talk about this in just a minute. But before we go on, let me, I do want to apologize and try to clarify a little bit. We 3D Molecular Designs released a line of student modeling packs last summer to have an option for students to be able to model individually. And so we tried to come up with something that could take the place of a lot of our kits. And so the one that we shipped some people, and we'll be shipping all of you who are participating in this session, is our nucleotide student modeling pack. And that's essentially just those foam nucleotides. We weren't able to create something that goes along with all of these kits. And actually this kit that Tim just showed you now, we've just started accepting orders in the past month. And I think all of our orders on our first kits, I'm hoping we'll be shipping everything out on Monday. So there isn't an at-home version of this. And so, but we were wanting to come up with some activities that people could do with those foam nucleotides that go along with this. So if that's a little bit of an explanation why you got these nucleotides, and the kit doesn't necessarily have anything to do directly with it, but it all has to do with those spacer sequences. Sorry, the repeat sequences. So, and I don't know if other questions have come up while I've been talking.
I can.
Can you see those?
Yeah, the question about how does this endonuclease recognize so many different spacer sequences? Well, again, that's because this system, this CRISPR system, it has been constantly at work in this bacteria over the lifetime of this bacteria, such that anytime a virus tries to infect it and this bacteria cell survives, then because there's an active CRISPR system, a little piece of that has been incorporated into the CRISPR array so that you can generate these separate surveillance complexes, which are looking for purple or tan colored or blue colored viruses. So, so again, we will go into more details as to how this Cas9 protein binds to the double-stranded DNA and interrogates the DNA with its spacer, and then if it's complementary, it cuts it.
Okay, we have just a few more minutes. I wanted to share just a little bit more information. There, we have up on the screen now some what we call guiding graphics that help describe what is happening in this process. So we aren't going to necessarily talk through all of this now, but this is essentially what Tim just went through with you. And then this third slide here is really what we're going to do in that next section. And there's even some drag and drop Google slide activities, which if I stop sharing, Chris may be able to share the screen and do some dragging and dropping and show you just some of the things that we can do associated with that. And so she's, so you can have your students actually model this either at home, virtually, either instead of using the kit or with the kit, but this is only available to people who've purchased the items. Okay, oh, and we did not get to, as we move on, Chris, there's, guess there are a couple of other slides we didn't get to about the CRISPR yogurt.
Oh.
What was that?
Yeah, this is something we don't really have time to talk about in this short session, but again, this is the reason you need to come to some of our summer courses, because this is Rudolph Barangu, who, and he's with his lab group there at North Carolina State University. But Rudolph, as a younger man, he must have been a kid when he did this, he carried out the definitive experiment in 2006, 2008, that finally proved what people were beginning to suspect, and that is that this CRISPR system was in fact functioning as this adaptive immune system in bacteria. So if you were to come to the summer course, we would talk a lot about Barangu and his classic paper that actually proved without a doubt that this was functioning as an adaptive immune system, and that every time he challenged a bacteria with a new virus, he was able to find a 32 nucleotide long insert in this CRISPR array. So beautiful piece of work. And it gives your kids an opportunity to look at data and then interpret that data. So it's another way to make this system more meaningful, more memorable to your students. So we'd encourage you to do that.
Okay, and great. We want to thank you all for attending today. And let me see. So if you have any final questions, post them in chat. We will stay for a few minutes after the session and answer as many as we can. A couple of quick reminders, make sure you've completed that link for your free student modeling pack. And you can use the code 3DMD Engage 21 for 20% off most of our products. And please complete that session evaluation by clicking on the link that Rachel's posting in the chat. And I hope that you'll join us for the next session called CRISPR-Cas9, a powerful new tool in editing the human genome. And then we'll really be showing you what's going on in using CRISPR for some of that editing. And then after that, we actually have our water kit session. Okay, any other questions that have come up? So, Katie, I love that you set up the lesson so you can discover CRISPR. Great, thank you, Katie. Well, then if nothing else pops up, thank you all for attending our session. We need to wrap up so we can get set up for our next session. We'll be ending the session in just a minute. You can leave anytime. Please complete that evaluation form and join us in the next session. Thank you again.